I'm Dr. Amit Bhattacharya from Department of Zoology, Ramjas College, University of Delhi, and today I will be presenting a topic which is biotechnology, tools and techniques. In this topic, I will be covering upon some of the basic tools and techniques which were developed over the years in a timeline way. So how exactly these tools helps in the diagnosis, development of various drugs, vaccines over a period of time. Now the biotechnology, the name suggests, it's basically amalgamation of the biology and the technology. Now this biotechnology helped the humans over a period of time to develop various transgenic crops, drugs, vaccines, diagnostic tools. So these are helping the humans to develop and detect the various diseases which were earlier tough to detect. Now initially the biotechnology term was given or used for the first time in 1917 by an Hungarian engineer. His name was Karl Erke. Now he was the one who first used it, but the exact and the precise definition of this particular term was ignored or not known at that particular time. Subsequently, in the year 1961, the Swedish microbiologist, he used and recommended the title in a more precise and specific way what exactly the biotechnology is, and subsequently the development and the change of the forms was published in the Journal of Microbial and Biochemical Engineering and Technology. Now, the biotechnology, as I told you, is basically an amalgamation of biology and technology in which the various fields of biology and the technology related are amalgamated, which includes the molecular biology, microbiology, biochemistry, immunology, genetics, genetic engineering, cell biology. So they all funnel into the development of the molecular biotechnology, which helps in the development of the crops, newer crops, drugs, vaccines, diagnostics, tools and the livestock. Now we will be taking up some of the important discoveries which were done in the field of the biotechnology and how it became one of the most emerging areas in technology right now. Now, the first the development took place in the year 1937. Now, in this one, the Nobel Prize was given to a scientist. His name was Tiselius. Now, what he developed was the moving boundary electrophoresis. Now, this particular electrophoresis helped in the segregation or the separation of the serum proteins from the sample. In this, if we see the picture over here, there is a serum sample protein buffer which is there, and on the either side there are two electrodes which are being loaded. Now as these electrode or the charges are switched on, what happens is there is a movement of this particular buffer depending upon the charges over there, and these proteins are then separated. Now at one point we can see there is a ascending boundary, while on the other part there is a descending boundary over there. Now this further gave rise to one of the very important technique which was later on known as the gel electrophoresis. Now in the gel electrophoresis we separate out the DNA, the RNA samples on a gel. So what we do is we load them on a gel or a cassette which is there. So we can see over here the instrument which is shown over here. Once it is loaded on the cassette, then the electrodes are switched on. So what we know is the DNA and the RNA are negatively charged. So what happens is they start moving on the direction towards the positively charged. Now in this movement process they get separated within the gels which contain beads. So the smaller fragments comes faster, so they move faster and they are at the bottom position, while the larger fragment remains near the wells only. Now this particular thing can be then analyzed or the quantified with the help of the use of the fluorescent probes or the radioactive dyes over there. Now if you see over here, there is a ladder which has been run. This ladder is basically helping us to understand what is the fragment size. So there are standard known ladders which are there, which are run along with the sample wells. So they help us to know that it is 100 base pair or 200 base pair or 300 base pair, what is the fragment size in these particular movement. Now subsequently in the year 1962, the Nobel Prize in Physiology and Medicine was given to 3 scientists. They are the one who discovered the structure of the DNA over there. So Watson, Crick and Wilkins were the one who were given the thing. Now if you see the right hand side picture, this is the picture or the page of the Nature journal, in which for the first time the discovery was published over there. So we can see a DNA double helix structure. So the discovery of the DNA played a very important or foundation development process in the biotechnology field. Subsequently, there were the discovery of the antibodies, which we called it as immunoglobulins over there. So the people who discovered them were given the Nobel Prize in the year 1959. Now we know that the antibodies or the immunoglobulin has got 2 major chains. One is called as the light chain, the other one is called as the heavy chain. Now the heavy chain has got a variable region and a constant region, while the light chain also has got a variable region and a constant region over there. Now the 2 heavy chains are attached to one another with the help of a disulfide bonds, while the light chains are also attached to the heavy chain with the help of the disulfide bond. Now the discovery of these antibodies or the immunoglobulin played a very important role in the development of the diagnostic tools. We will see some of the very important diagnostic tools, how they changed the developmental process and the screening process in detecting the various diseases which were not the scientists or the doctors were unable to detect in the previous phases. Now in the 1970, the scientists got a Nobel Prize for the discovery of the restriction enzymes. Now the restriction enzymes many a times are known as the molecular scissors or the biomolecular scissors. Now what they do is they recognize the specific site within the DNA and they cut the DNA at those particular site. Now this particular cut and the ligation help the scientists to cleave out the fragments which is not required. So over here if we see enzyme which has been shown, which is known as the EcoR1 enzyme, which has got a restriction enzyme site GATTC. So it recognizes this palindromic sequences. The palindromic sequences means the sequence which reads the same from the forward and the backward sequences. Now the palindromic sequence is recognized and it binds to this palindromic sequences, producing a staggered end. So we can see there is a overhanging regions which are being produced, and a single linear DNA is now cut into 2 fragments in which there is a staggered end. Now this particular thing helped in the recombinant DNA process also. Now if we see over here, there are 2 types of major end cleavage which are being done. One is called as the staggered end cleavage, in which the ends are staggered, while the other one is called as the blunt end cleavage. The enzyme over here is called as the Hind2. Now if we see over here the staggered end, there is a overhanging region over there, while in the blunt end there is a blunt end which is there. So depending upon the site what type of enzymes is required, there are different types of restriction enzymes which are there. So it is being said that about 3,700 type 2 restriction endonucleases are present, in which about 250 different recognition sites are being recognized by these restriction endonucleases. So we can imagine how useful these particular enzymes can be. So they will be helping in recognizes the various sequences, and then the sequences can be cut accordingly depending upon what kind of ends we need to create, either staggered or the blunt ends over there. Now these are some of the examples which are shown over here. these examples show the various enzymes over there. So we can see the EcoR1, Hint3, BAMH1 like this one. So we can see some of the enzymes, they produce the staggered end, and some of the enzymes produce the blunt end like the He3, which is present at the bottommost part. So we can see there is blunt end which is produced over there, and rest of the enzyme produce a staggered end depending upon the sequence in which they cut. Now, these particular enzymes were isolated from the bacterial system. Now, the question comes is, why does the bacterial system requires these restriction enzymes within their system? Now, because the bacterial system doesn't have such an advanced level of immune system and these clearance system what the humans and the other developed organisms have, so what they have developed is the restriction enzymes. Now what they do is, suppose there is a foreign DNA which enters into the bacterial system, they immediately recognizes the cleavage site within that thing. So suppose if we see over there, there is a EcoR1 restriction enzyme site which is present. So we can see over here the GAA-TTC site is recognized by the bacterial system restriction enzyme. So this particular enzyme binds over there and it cuts or cleaves this particular site, thereby disrupting the foreign DNA which has entered into the bacterial system. Now, how exactly the bacteria saves its own DNA from this restriction enzyme? The answer is very simple: methylation. There are certain methylase enzymes which is present within the bacterial system. What they do is, these methylase enzymes, they methylate specific sequences which are being read by these restriction enzymes. Now, these are then methylated so that the self DNA is not restriction enzyme digested by the enzymes which is present in the circulation. Thus, the bacteria prevents the destruction of its own DNA with their restriction enzymes just by a simple process of methylation. Now, we take an example how exactly these restriction enzymes, cleavage site or the restriction enzymes were used for the detection of the disease. One of the very important disease which is there, which is known as the sickle cell anemia over there. Now we know that in the sickle cell alleles which are present in the beta globin gene or the hemoglobin gene over there. So if we see over here, there are certain restriction enzyme sites which are present in the normal beta globin genes. So DAD1 enzyme is the one which is present. So if we see, there are two restriction enzyme sites which are present in the normal beta globin gene over there. But what happens is, we know that in the sickle cell anemia, there is a point mutation due to which one of the restriction enzyme site within the normal beta globin gene gets disrupted. So it is removed from that particular site. Now, if we digest both the normal as well as the sickle cell anemia mutant beta globin allele, what will happen is, in the normal there will be 3 fragments which will be produced because there are 2 restriction enzyme sites within them, while in the case of the sickle cell mutant form there will be 2 fragments which will be produced because one of the restriction enzyme sites have been mutated or changed. So the enzyme cannot recognize the site, it can recognize only one site within that particular beta globin gene or the allele. So when this particular sample is run on the electrophoresis over there, we can see that the restriction fragment analysis helps the scientists to distinguish between the normal and the sickle cell anemia of the beta globin gene over there. So what happens is, in the case of the normal alleles, there are 3 fragments which are produced. So there are 3 fragments which can be easily seen in the restriction digestion as well as in the electrophoresis, while in the case of the sickle cell anemia, because there were 2 fragments which were produced, so these 2 fragments are recognizable over there. So with the help of the restriction enzyme analysis as well as the electrophoresis, scientists can easily decipher out and quantify the difference between the normal and the sickle cell anemia samples over there. Now, the next important assay which was developed was the development of the enzyme linked immunosorbent assay, which is many a times referred as the ELISA assay. Now, this was developed in the year 1971. So, what this assay did is it can easily detect in the sample the amount of proteins which is present or the amount of antibodies which are present. Now, depending upon the procedure or the detection pathways, it was detected into two different forms. One was called as the indirect ELISA, the other one was called as the sandwich ELISA. Now, in the indirect ELISA, it detects the presence of the serum antibodies against a specific viruses. Now, this indirect ELISA is being used for the development of the diagnostic tools for the HIV infections or to detect whether the person is suffering from the HIV infection or not. So what it is being done is the antigen, the wells are being coated with the antigen, and we know that if a person is suffering from a viral infection, for example the HIV infection, there will be production of the plasma antibodies or the primary antibodies within the body. Now, when the samples are taken and from the sample the serum is extracted and loaded onto these particular sample wells, if the person is suffering from the HIV infection, the primary antibody will go and bind to the antigen coated wells which has got the antigens present within them. Now, subsequently, the secondary antigen are loaded on the well which contains specific enzyme which can change the color of the substrate into a colored product. Now, with the help of this change of the color, we can easily detect within the well whether there is a presence of the serum antibodies against HIV or the human immunodeficiency virus is present or not. Now, the next important ELISA which was developed was called as the sandwich ELISA. The sandwich ELISA help in the detection of the presence of the antigens, especially in the case of the malarial antigens for the detection of the falciparum in the blood samples. Now, what the scientists did is they had the antigen well coats over there which has got antibody coated in the wells. Now, if a person is suffering from a malarial infection, we know that there are certain kind of antigens which are circulating in the blood circulation because the scisons which are ruptured, along with the rupturing, there are certain antigens which are released in the blood. Now, the scientists can easily detect these antigens from the blood samples. So, the blood samples are taken and then they are centrifuged and loaded onto the wells. If the person is suffering from the malaria infection or the falciparum infection, the antigens for the specific falciparum will be present and they will go and bind to the antibody coated wells which has got antibodies present within them. Now, subsequently, the secondary antibodies are loaded which has got specific enzymes present to them which can convert the non-colored substrate into a colored product or the colorless substrate into a colored product, and the wells can be easily detected. So, with the help of these particular techniques, the scientists can easily detect the presence of either the serum antigen or the presence of antigens within the circulation. Now, if we see over here, this is a two-step process which is shown over here, how exactly the medical diagnostic tools are using the ELISA for the detection of HIV infection within the AIDS patient. Now, the blood samples are taken in and we know that if the person is suffering from the HIV infection, the serum protein will have antibodies present in them. So, those are loaded onto the ELISA cassettes over there, and in these cassettes we have got the indirect ELISA which is followed over there, and subsequently what happens is if the person is having a infection, what we can see in the test well, there will be a line which will be shown, as well as there will be a line in the control. So control is the normal one, which is shown both in the infected and a non-infected individual. But in the infected individual, if the test well shows a line, it can easily detect that the person is suffering from the HIV infection. Now, to further quantify how much the infection is, we can go for a tighter quantification. So, in the tighter quantification, the presence of the serum antibodies from the blood against the human immunodeficiency virus is detected. So, we can titer out or dilute those particular samples various way. For example, if we see 1:2, 1:10, 1:100 dilution is shown over here, and we can see in the patient B, C over there, there are presence of yellow products which are being shown. So we know that there is a high titer or the presence of the serum antibodies which is present in the patient C as well as in patient B. So the titer volume tells us or gives us an idea how advanced level of HIV infection has been there in the patients over there. Now, these particular techniques, like the development of the restriction enzymes and the other techniques, they give rise to the development of the recombinant technology process. Now, the recombinant technology process, as the name is suggesting, it is recombining the DNA technology process. Now, in this one, two of the scientists played a very important role, Boyer and Cohen, in the development of this particular process. Now, in this process, in the year 1973, they started developing this process, how exactly a plasmid DNA or a cloning vector can be used to transfer a certain fragment of gene or the target gene into a host system, which can then replicate, transcribe and translate to produce the proteins, and these proteins then can be used for the further processing. Now, if we see this diagram over here, this diagram shows how exactly the recombinant DNA technology is following up over there. Now, in the recombinant DNA technology, what happens is, suppose there is a target gene which needs to be incorporated into the plasmid vectors. So, the plasmid vector is shown over there, which has got the multiple cloning sites present within them, and then there is an antibiotic resistant gene section which is there, and the origin replication site which is present. Now, with the help of the restriction enzyme, one type of restriction enzyme, both the target gene as well as the cloning vectors are cleaved. So what happens is, suppose there is a staggered end which is produced in both the target DNA as well as the cloning site, then they are ligated. So with the help of the staggered ends, they can easily get ligated into the cloning vector. We develop the cloning, recombinant cloning DNA over there. Now this recombinant DNA clone vector is then transferred into the host system with the help of the various processes, which include the calcium chloride or the electroporation process. And now this recombinant cloning vector is present within the host system, where it utilizes the host system to develop and transcribe and translate to produce the proteins over there. So the recombinant proteins are produced there, which can be then further purified and isolated. Now, this particular technique was initially used for the development of the human insulin, which was many a times referred as the recombinant human insulin. So in this technique, what it was done is, the insulin gene were isolated. It was cloned with the cloning vector over there. So we can see over there the restriction enzymes were used, and then this insulin gene was cloned onto these restriction digested cloning vectors. And once this cloning vectors or the recombinant cloning vectors were isolated and they were checked, then they were transferred into the host system, which is generally the yeast host system or the various other host system, where it utilizes the host system mechanism to transcribe and translate. Once the translation has been done, the proteins or the insulin proteins which it's produced over there, they are isolated, then they are further processed so that a proper human insulin is formed over there, and subsequently what happens is these particular human insulins are then purified and packaged into the bottles, which can be then used by the patients who requires the injection of insulins or are suffering from the diabetic condition. So this recombinant DNA technology played a very instrumental role in the development of these particular recombinant protein molecules or the DNA forms. Now this not only gave rise to the insulin, subsequently there were growth hormones which were produced and the various other things which were produced. So now there are various recombinant protein forms which are there, which are being using this recombinant DNA technology in the labs to produce these human forms of the thing. Now in this part of the lecture we discussed about some of the basic tools which were developed over a period of time in the biotechnology and how these techniques were used in the diagnostic tools for the further diagnosis and the screening of the patients for the things in a better and correct way. So with this note I come to the end of the lecture. I will continue the next part of the lecture. Thank you very much.